Okay, so welcome everyone to the first webinar of the series on implementing targeted next-generation sequencing for detection of drug-resistant TB in high-burden countries, from recommendations to reality. This series is being organized by the Stop TB Partnership and FIND in collaboration with USAID, UNITAID, the Global Laboratory Initiative, and the New Diagnostics Working Group. My name is Wayne Van Gemert, and I'm a Diagnostics Technical Officer at Stop TB.
And I'm Anita Suresh, head of the sequencing unit at FIND. Our first webinar is focused on the newly published WHO recommendations on targeted next-generation sequencing, or tNGS, implementation considerations and resources, as well as country experience. So representatives from Kyrgyzstan and Eswatini will be sharing their experiences and lessons learned using tNGS for surveillance and clinical decision-making. In future webinars, you'll hear from manufacturers on their tNGS solutions, ongoing efforts to update and improve the assays, as well as more country experiences and operational elements.
A couple logistics details. So this webinar is being recorded, so we'll be sharing the link afterwards, as well as the presentations. If you haven't seen it yet, there is an option for French and English interpretation. So if you click the globe icon at the bottom of the screen, you'll get this in French. And we encourage you to take advantage of the Q&A box to write your questions. We do have some time scheduled at the very end of the webinar for Q&A, but hopefully the speakers will be able to answer in writing some of the more straightforward questions. So without further ado, I would like to introduce the Deputy Executive Director of Stop TB, Dr. Suvanan Sahu, who will share with us some welcome remarks. Over to you, Sahu.
Thank you. Good morning, good afternoon, or good evening, depending on from where you are joining this call. Greetings from Stop TB Partnership and a warm welcome from Stop TB Partnership. This is a webinar, as you know, on targeted next-gen sequencing. And as Wayne informed us, this is being organized by Stop TB along with FIND and in collaboration with the USAID, UNITAID, Global Laboratory Initiative, and the New Diagnostic Working Group of Stop TB Partnership. It is exciting and great to see so many people connected. I see that 238 people connected, and it's growing. The numbers are growing as we speak. Actually, we had registrations of 900 people. So that shows the great interest in this topic in the TB and in the diagnostic community. So great to see this and very excited to look and we look forward to the discussions, not only in this webinar, but we hope to make it a series so there will be more on this coming. So it is very clear that we have a big diagnostic gap in our global fight against TB. Millions of people are not diagnosed for TB each year. And if you look at drug-resistant TB, we need better diagnostics and better access to diagnostics. WHO estimates each year 410,000 drug-resistant TB, people suffering from drug-resistant TB. But if you look at the people who are diagnosed and treated in 2022, only 176,000. Over the years, we have never reached about 50% of the estimate. That's a big, big diagnostic gap. This is a challenge for all of us. Some of this challenge can be addressed by innovations in diagnostics, and this is something that we are discussing today. And diagnostics, we must remember, do not self-implement. So we have to provide access. We need to scale it up and provide access to people. The more people benefit from the new diagnostics, the more people we can look forward to diagnose. We have to remember that 2023 September, the countries committed in the UN high-level meeting in the UN General Assembly to treat 1.5 million people. 1.5 million people between 2023 to 2027. 1.5 million people with drug-resistant TB. We have to do our best to achieve that target. If we continue business as usual, we will not even reach half of that. So we have to scale up diagnostics, and particularly on drug-resistant TB. For years, we have been wanting to have better diagnostics. And I must say, in the last few years, a lot of innovations have happened. We also have new drugs now for treatment of TB. We are all excited because treatment of TB has now been, treatment of drug-resistant TB. We are excited because it has become as simple as drug-susceptible TB, just 6 months. And not very complex, few drugs, all oral. But we must also realize that as these new drugs come, we should have the capacity to also test for drug susceptibility, for resistance to these new drugs. We know about bedaquiline, delaminid, linezolid, pretonamid. There are tools currently available that are focused on the older drugs for DST. And these tools are simpler to do. They are rapid molecular tests. But when we look at the whole spectrum of drugs that we have today, we have to rely on something like this next-gen sequencing technology to get the drug susceptibility testing known for all drugs. This area is also rapidly progressing. We saw how during the COVID pandemic, countries developed capacity on genetic sequencing. We must build on that capacity. Today, we have capacity in Africa, in Asia, in low-income countries to do that. If that was done for COVID-19, why can't we do it for TB? We can build the systems further up and scale it up for TB diagnosis. So this is a great opportunity. I think we must do our best to scale it up, first get the resources, but also to get the expertise and the capacity to scale it up. And what actually will save life is not just providing the diagnostics, but providing access to people to benefit from the diagnosis. So that's all from my side. Very excited again to see the numbers again growing up of people connected. So look forward to a very exciting discussion. Over.
Thank you, Sahu. I would now like to introduce Dr. Marta Fernandez Suarez, who's the Chief Technology Officer of FIND. Over to you, Marta.
Thank you, Anita. And good morning, good afternoon, and good evening to everyone. I join Sahu on welcoming all of you to this webinar on the use of targeted sequencing for TB. As many of you are aware, and Anita mentioned this in her opening remarks, the recent WHO recommendations really highlight the potential of targeted sequencing to change the way we test for drug-resistant TB. And it is now the time to ensure that we realize that potential. So today in this webinar, we would like to present evidence to demonstrate of the potential that we see. And there is a lot to do, right? We just discussed by Sahu, to enhance implementation, to integrate into the algorithms, to do a scale up. But it is already happening. And today we are honored to have some country representatives from Kyrgyzstan and from Eswatini who will discuss already their real world experiences and their lessons learned. There was a huge increase in sequencing capacity during the COVID-19 pandemic. The install base grew tremendously in many countries around the world. And it is now the responsibility to ensure that this capacity is being used. And TB drug resistance offers a great opportunity to fill the gaps that we have seen over the years. So with that, I just want to thank you all for joining us. And I want to thank our partners at Stop TB, at USAID, at UNITE, GLI, and the New Diagnostics Working Group for collaborating with us and making this webinar possible. I hope you all enjoy.
Okay, thank you, Marta. So we'll move on now to the first presentation, and I would like to introduce Dr. Nazir Ismail, who is a senior advisor on TB diagnostics to the WHO Global TB Program. Nazir will be speaking on the recent WHO recommendations on tNGS. So the floor is yours, Nazir.
Thanks a lot, Wayne. Let me just get my slides up. It is not in full screen, probably, right?
Not yet.
1 second. Yeah. Right. It should be full screen now.
Yes, go ahead.
Okay, brilliant. Thanks a lot, Wayne. And good morning, good afternoon, and good evening to all of you, and thanks for joining. So I'll cover briefly the new recommendations by WHO on targeted next-gen sequencing. I just had two slides just as an introduction, just to make sure that people that are not familiar with sequencing, just as an intro. But to begin with, the tNGS guidelines come up after a series of different recommendations, primarily focused on a real-time PCR type of platforms in the more recent past. And sequencing is quite a significant shift in the type of technology being used for drug resistance detection. So just to give some context, we've historically used multiplex or specific PCRs that are focused on a single drug and a single gene, and usually a hotspot in it. So, for example, RRDR, we focus on 81 base pairs in the hotspot region. Whole genome sequencing is on the other extreme, where it's a 4 million base genome, and sequencing could cover the entire genome. And what we're focusing today is really about targeted next-gen sequencing that just targets specific genes of interest, primarily focused on drug resistance targets. And to give you an idea, for our POV genes, about just over 3,000 base pairs. So the targeted injection sequencing is really drug specific and focusing on relevant genes of interest. And I think Wayne will Andrea will cover this a bit later on. But sequencing, as I indicated, is a very new approach, starting off from DNA extraction, and depending on the methodology that's used for the DNA processing, library preparation, amplification. And while we use the term sequencing, it's part of a process. And that sequence generates files, and those are interpreted usually by the mutation catalog described by WHO. So it's a fairly complex process, but quite an advanced way of detecting drug resistance. So specifically on the targeted next-gen sequencing in terms of the WHO recommendations and the class, this has been defined as a product that uses massively parallel sequencing to detect resistance to TB drugs. And specifically for the purpose of this WHO guidelines about a product that that has a procedure that starts from a clinical sample and ends up with an interpreted result at the end, obviously following a standardized interpretation of mutations. So the WHO's GDG process started back in 2022, where the topic was raised. A commissioning of a series of systematic reviews were undertaken to answer this question, both looking at published and unpublished data. And these reviews covered both the accuracy information, but also economic and qualitative evidence, including values and preferences of individuals. The meeting was convened in May 2023. Where the group reviewed the available evidence and went through a standardized grade process. A rapid communication was issued, and the full guidelines were issued now in March 2024, just before World TB Day. And what I'll be covering is the key findings and recommendations from there. So what I'll be presenting is available on the web, and has been incorporated into the consolidated guidelines, which is the third edition, the latest edition, and also in the accompanying operational handbook, and the hyperlinks on the screen in front of you. In terms of the targeted next-gen sequencing, following the public call and the review of evidence available, 3 products were considered within the a guideline review process. The first one was by Dplex, which is has a product called MITE-TB. And for the guideline process, the performance criteria for the drugs listed were met and therefore included in the evidence synthesis. The second group of products, which was from Oxford Nanopore, in a product called MPOR-TB, with the reduced number of drugs that met criteria. You'll see the absence of, for example, bedaquiline in the current kit that's listed here. And then TB-SEC, which was a product from Shenting Biotech, which is only included for one drug, and it was really primarily driven by the fact that the reference standards included for their data generation were as per required specification. But there will be opportunities for these other 2 products to submit their data to WHO for further evaluation and consideration for the other drugs as well. and obviously any other future products that come up as well. So specifically in terms of looking, and I'm just limiting it to the individuals with rifampicin-resistant TB, the overall accuracy was really good for drugs like isoniazid, levofloxacin, moxi, parazinamide, and ethambutol, with very high sensitivity, it's above 95%. And for other drugs of great interest like bedaquiline, linezolid, clofazimine, the accuracy was deemed acceptable. Also for drugs like amikacin and parazinamide. So there was a difference in performance, but the group appreciated the fact that this was a more rapid test and addresses the need. In terms of specificity, largely the accuracy of the test was really good, with specificity above 95% for all drugs except streptomycin. But even in that case, the reference standard was most likely a problem. In terms of the economic and qualitative findings, this was very complex. We know that targeted next-gen sequencing is not cheap, and the cost-effectiveness modeling that was performed had variable findings. It really varied by the prevalence of resistance in a specific given context, what was being used as the current standard of care, and the potential of this new technology to impact loss to follow-up. Important to appreciate that where settings where there isn't as robust a testing system for drug resistance, cost effectiveness would obviously be better than setting up phenotypic DSC on a large scale. What is also quite clear is that the cost would vary based on the implementation approach. So often batch testing is used for targeted next-gen sequencing and would really be volume dependent that would impact on cost effectiveness. And then lastly, I think as we've heard earlier on, utilizing the technology across multiple diseases. We know COVID had scaled up a lot of product capacity in terms of sequencing, and that would reduce cost burden. In terms of the qualitative aspects, the targeted NGS was deemed acceptable and implementable in routine programmatic settings, despite the inherent complexity. And I think that it needs to be appreciated that it's not a very simple test like EXPERT, so it's a major shift in how it needs to work, but it does have advantages and would require technically a level of technical complexity, both infrastructural and having the skilled staff to perform these tests. And what is also appreciated was that different implementation models and algorithms would need to be investigated to identify the most effective way of deploying the technology. So having said that, the group made a positive recommendation on the use of targeted next-gen sequencing. There were two recommendations. The first one relates to the use of targeted next-gen sequencing for bacteriologically confirmed TB. So it would also include people with rifampicin susceptible TB. It's a conditional recommendation and covered drugs like rifampicin, INH, the fluoroquinolones, parazinamide, and ethambutol, all relevant for the treatment of first-line therapies. What was also made clear in the remarks related to these recommendations is that priority, specifically in this group, would be for higher risk individuals, where the test should be prioritized, and that would be people that are not responding to therapy or remain smear positive, those with prior treatment history. So all the risk factors that drive resistance as such. And this is a conditional recommendation, as I indicated. So would be useful as a secondary test where there might be discordance results, so patients might have tested susceptible, and people are concerned. The second recommendation is around the population for rifampicin-resistant TB. I think this is probably the most prioritized target group, at least for the early implementation. And here there was also a positive recommendation for its use in this population on respiratory samples to diagnose resistance, and the drugs include all that were considered. So INH, fluoroquinolones, bedaquiline, linezolid, clofazimine, parazinamide, ethambutol, amikacin, and streptomycin. And this would be rather than culture-based DST. It is a conditional recommendation, and I'll go through some of the further considerations. Again, similarly to the previous one, prioritization should be given to those at higher risk of resistance. And those, again, would include people with smear-positive disease after 2 months, those who had previous exposure to the drugs of interest, had contact with a known drug-resistant case, and also those that had pre-XDR TB, meaning that they have fluoroquinolone-resistant TB, all higher-risk individuals. In terms of the implementation considerations, which need to be read in conjunction with the recommendations, so very clear that regulatory approval needs to be achieved before implementation for any product that's considered. There would need to be scale up in terms of specialized skills and infrastructure. And then what is very clear from the group's decision was that, at least for the first recommendation, targeted NGS does not replace existing rapid tests that are more accessible and easier to perform, particularly for drugs like rifampicin, INH, and fluoroquinolone. So it's not really, we should be using those technologies that are easier and cheaper. However, where it is justified and needed, this could be as an alternative option for prioritized populations. What is also noted in terms of the technology is that the indeterminate rates were really dependent on bacillary load. So those with higher bacillary load, either semi-quantitative, high or medium on the expert or smear positive, the test performed well. However, the indeterminate rates were higher in those with low bacillary loads, and therefore prioritization obviously cost and feasibility dependent. So moving on to the more tricky aspect is the performance of the technology for detection of bedaquiline, linezolid, and clofazimine resistance, which I think is probably the most highest priority, I think, currently as we move forward. It's very clear that the performance of this test, the test for these drugs was suboptimal. So roughly around 68% for bedaquiline and roughly similarly for linezolid and clofazimine. And for bedaquiline, the specificity was slightly lower than for others. So the group highlighted the important need to look at pretest probabilities in trying to appropriately use these technologies. And then what I also appreciated that the test does not test for pretonomid at present, and therefore culture-based DST is also required for that drug. I've pasted here the evidence table just to give some context in terms of the next slide that I'll go through. But we need to appreciate also for the current status in terms of resistance to many of these drugs like bedaquiline, linezolid, and clofazimine. The relative prevalences are fairly low. And we also need to take into cognizance that the performance of the test were suboptimal, and therefore nuanced approaches for how it's implemented are needed. And just to be quite clear, I put the table of bedaquiline in front of you, so you see what's the sensitivity and specificity for bedaquiline. And looking at a simulated 1,000-patient scenario with a pretest probability of 3%, which is likely to occur in many settings for bedaquiline, where it hasn't really been widely used. We know that in places where it is widely used, the prevalence would increase to above 5%. And in that case, obviously, the risks are high, and therefore the nuances may not be as required. But just to be clear on this table here, and I'm going to spend a few minutes on this just to be clear. So if you're looking at 3% pretest probability, among those with true positives, you'll end up with about 20. But in that same group, the amount of false positives would be 29. So you'd end up with more false positives than true positives. And therefore, again, depending on the regimen that you're looking at, if BPL-M versus a 9-month regimen, decisions are quite critical in terms of who you select and who do you deprive of the newer regimens. And then secondly, in terms of the false negatives, while 10 out of the 30 positives would be missed, but if you look at it on the broader perspective in terms of false negatives at that low prevalence, among those that test negative would be the 10 false negatives and then the 941 true negatives. So largely the negative predictive value would be very high in low prevalence settings as such, at least in this context. So this is a table, and I'll spend a few minutes on this as well. We had issued the operation handbook for this table, but there were a few, I think, layout issues that might have led to some confusion. And the table is being updated. So this is a draft updated table, but would be something similar in terms of the corrections that are being implemented. And just to explain the situation, I think it's a fairly complex situation, and what we've tried to do and to make the application much more clear and easier is we have a column that says initial regimen. So you'll see BPL-M, and then on the right-hand side, you look at the 9-month regimen and individualized regimen. So that's kind of your starting block. The second is the drug of interest. So with targeted next-gen sequencing, you can detect drug resistance to multiple different drugs. So you have all of that information at one time, and therefore allows you to utilize that information to triage and decide which patients should be managed in which way. The next column is the targeted NGS results. And here you'll see, based on the recommendations and implementation considerations, stratification is done in terms of risk category, so low risk and high risk. And the high risk, obviously, those that have prior exposure, that have additional fluoroquinolone resistance, and those where the prevalence is above 5% is what is defined in the operational handbook. And the last two columns are really how to manage those results as such, in terms of whether phenotypic testing is required or not, and how what regimen modification should occur, whether that should mean a person has a change to an individualized regimen, or could treatment be modified or not. So it's a fairly complex table. I don't think I have enough time to go through it as such, but maybe just a few things to highlight. So in terms of bedaquiline particularly, I think that's been of the greatest interest. So if you look at the 9-month regimen, if any resistance to fluoroquinolones, bedaquiline, linezolid, or clofazimine is identified, that really requires a change in regimen, except for linezolid, where there are some nuances. And as such, the results from the targeted next-gen sequencing would be directly applicable and interpreted as is. For the BPL-M regimen, this has some nuances, and that's what's on the left. So in situations where there is resistance detected and the risk is low, there are considerations of when patients would continue on therapy, waiting for phenotypic confirmation, appreciating the slightly lower specificity. But obviously, in a high-risk case, that's regarded as a true resistance and managed accordingly. And similarly, for bedaquiline results with susceptibility, often these would require continuation on BPL. But if it tests susceptible and there is risk for resistance, then phenotypic cultures and close follow-up are therefore required. So just to clarify, this is really meant to address a complex situation where we appreciate there's some optimal performance for some of these drugs. We know that there are variable prevalences, but it is emerging. We also appreciate the importance of the new regimens, the 6-month regimens that are really potent and beneficial to patients, and recognizing that there aren't many alternative therapies if these new regimens do not work as such. So in conclusion, I think the introduction of targeted next-gen sequencing marks a major shift in the diagnosis of drug-resistant TB, addresses important gaps for new and repurposed drugs, but also recognizes less optimal performance. The cost and feasibility are potential barriers, but also offers important opportunities post-COVID. And I think many countries have sequences available and have used sequencing for COVID. The best implementation approach is yet to be defined, but will likely be context dependent. And lastly, more evidence is needed on the new drugs and the impact of targeted NGS on patient-important outcomes. Thank you very much.
Thank you very much, Nazir, for breaking down all the details of this very important but also quite complex guidelines. I would now like to introduce Dr. Andrea Kabibe, who's a research associate at the San Raffaele Scientific Institute in Milan, which is also a WHO TB supranational reference lab. Andrea is also a member of the GLI core group, and he will be speaking on the tNGS implementation considerations and resources. The floor is yours, Andrea. Thanks.
Thank you very much, Anita, for the nice introduction. And hello, everyone. I'm going to share my screen now. And you should see in presentation mode, I hope. And
Yes,
here
it's clear.
Okay, great, great. So hello, everyone. And we are now going through key considerations to make and available resources when a program or a laboratory wants to implement programmatic use of tNGS. And some of these resources are coming from the work done by the GLI. For those who don't know, GLI is one of the 7 working groups of the Stop TB partnership. And it represents stakeholders and institutions that are involved in the laboratory strengthening with the mission to serve as a collaborative platform for the development and uptake of practical guidance and tools for building and sustaining high quality TB diagnostic networks. And more details on the activities of GLIs are available at this link and using the QR code here. The first documents that I want to show you with regard to the implementation of tNGS are the information sheets that are available as annex to the WHO operational handbook on TB, module 3 on rapid diagnostics. And these documents includes the info sheets that summarize the individual technologies that are in the class-based recommendation. So it's not limited to NGS, but includes information for any rapid diagnostics included in the operational handbook. And for this presentation, we will focus on the 3 tNGS products that have been recommended for clinical use. And I want to focus specifically on some implementation and operational considerations and to compare the 3 products. And the detailed and all the extended information on these are available in the info sheets. So in terms of implementation, the laboratory level that is recommended for use of tNGS currently are the centralized reference settings, because the requirement for infrastructures are high or moderate to high, and specifically high for the Illumina platforms and moderate to high for the Oxford Nanopore Technologies platforms. In terms of regulatory aspects, the GenoScreen product, Diplex MIC-TB, is marked CE IVD, and the Amport TB kit is research use only, and the TB-Sec kit has been approved by the Chinese regulations. All the 2 platforms, Oxford Nanopore and Illumina, are suitable for multi-disease testing. And this is a very important consideration to make a cost-effective use of NGS. In terms of procurement and supply chains, the companies are trying to ensure global access to their products through partnerships with bigger companies. And moving to the operational considerations, and I think here we are addressing some of the questions that have been raised during the first presentations. So the sample types that can be used for the 3 products are decontaminated sputum specimens. These can be decontaminated in different ways, inactivated in different ways, but the starting point is a sediment, basically. I want also to show you what are the storage and handling requirements for the 3 products. And you can see that there is a wide range of temperature to ensure to keep and store the different reagents kits for the NGS workflow. And this is one of the considerations to make, and that request a high level of infrastructures. Testing capacity depends on the products that can be, from 24 to 96 per kit. The time to detection is usually shorter for the 2 tNGS kits that are using the Oxford Nanopore technologies. Although with the GenoScreen technologies, you can have different outputs, different platforms, Illumina platforms that you can use, and meaning that the batching size can be higher than the other products. Moving to the key performance conclusions, Nazir has already showed you what are the recommended drugs for each of the companies. These will be updated in the future. However, so far, currently, the Diplex can also be used for detecting resistance to bedaquiline and clofazimine. In terms of heteroresistance, so the capacity of a kit to detect the presence of minor populations in the patient sample. This is reported by the companies to be 3% for diplex and 10% for the 2 kits using Oxford Nanopore. The limit of detection has been specified for the diplex and the TB-Sec kits. And the 3 products are also capable to detect non-tuberculous mycobacteria. And the performance has been specified for the GenoScreen diplex kit that is able to detect more than 100 mycobacteria species. I will now move to some additional notes to the implementation of tNGS that has been done by Nazir, and I don't want to repeat some of the information that has been shared by Nazir. However, I just want to stress that tNGS is suitable for use at central laboratory or NTRL level, and and that the priority will be given to samples that have a higher bacillary load. And this doesn't mean that the kits cannot be used for samples with a lower bacillary load, but we need to acknowledge a higher indeterminate rate when the starting bacillary load is low. If tNGS result is indeterminate, the tNGS test can be repeated with a fresh sample if the bacterial load is expected to be higher and to give a definitive result. The platforms are suitable for multi-disease testing, meaning that multiple pathogens can be detected on the same platforms using different kits and approaches, but meaning that if you ensure that the platform, the sequencing platform, is being used regularly on a weekly basis, continuously, this will also ensure you to be cost effective in the capital investment that is requested for the NGS platforms. And one specific consideration in terms of sample, the need of samples for tNGS testing, if molecular, so if NGS and phenotypic testing are performed in the same laboratory, collecting one specimen for these 2 different tests may be sufficient. Now, I want to show you the outcomes of a rapid review that was commissioned by the WHO to identify qualitative evidence on the use of tNGS, because this enables to understand well what are the current challenges in the implementation of tNGS at country level for programmatic use. So in terms of acceptability, what was perceived to be good things from the participants to the survey was the comprehensiveness of the tNGS approach, the convenience of using the patient samples, the rapidity in terms of turnaround time to get the tNGS results and report, and the opportunity to benefit from the NGS utility. In terms of challenges for feasibility, of course, there is the startup and setting up of the NGS infrastructures, and we will also hear this from countries that are implementing NGS. The high technical complexity of the test that requires specialized laboratory infrastructures and specialized training for staff. The supply chain, as distributors are not always available locally. And again, we will hear about this experience from countries. The need of data management and storage, because bioinformatic data are somehow complicated to be handled and stored. And in terms of values and preferences and equity, there may be some equity implication for access when deciding for a centralized versus decentralized placement of tNGS. And in particular, in countries where the burden of MDR is high, this could influence equitable access at centralized levels. Now, I want to share with you very briefly, a couple of country experiences in implementing tNGS with specific regard to the implementation challenges that has been observed by the laboratories and the programs while implementing NGS for TB use. So this is the experience coming from a USAID-funded INTP project through the support of the Stop TB partnership at the ICDDRB Institute in Bangladesh, to Dr. Banu and Dr. Udin. And the key findings from this pilot study conducted by testing samples with rifampicin resistance or rifampicin susceptibility are outlined here. The turnaround time was 3 to 4 days from sputum samples referred to the sequencing laboratories. The cost was around $150 per tNGS samples using the Illumina MySeq kit and the Dplex GenoScreen solution. And in terms of accuracy, it was observed that high agreement with phenotypic and genotypic DST for most of the key drugs, and in particularly an excellent specificity of tNGS results. In terms of the implementation challenges that the laboratory had for using regularly tNGS was the procurement of reagents and NGS equipment because there was unavailability of local distributors. So this process can take a long time. The need to upgrade laboratory infrastructures, so it was necessary some renovation, and the need of special training. And in terms of the impact of this pilot study at the Institute is that the tNGS data was shared with the WHO for raising recommendations for the use of tNGS. And tNGS was included into the National Strategic Plan 2024 to 2030 of the National TB Program in Bangladesh. Second example is coming from a German-funded project to the SRL in Borstel and implementation at the University of Namibia. Here, the implementation process of sequencing infrastructures at UNAM took 5 years total for the preparation, planning phase, and implementation phase, and it was usually delayed by the COVID-19 pandemic. And the approach adopted was to use the small ISEC platform from Illumina. In terms of implementation challenges, again, the need to upgrade and construct the NGS laboratory infrastructures. There was some issue with power interruptions, with the need to ensure a certain temperature in the sequencing room, unstable internet connections, and the need to build dedicated pre and post PCR areas, the need of special training, delayed supplies of reagents, and some issues with importation, because sometimes the process of customs clearance took a long time, and then shortages. So for this reason, as backup plans, some products were procured by the neighborhood countries or by the SRL in Borstel, but this of course led to delays in testing and also to an increase in total cost for testing. And then the need of sophisticated tools for data management, storage, analysis, and interpretation. However, capacity was built for regular use at UNAM, and NGS capacity was developed within the TB project, was used for emergency response to the COVID-19, so for sequencing of SARS-CoV-2. And trained personnel at UNAM trained other laboratory scientists from the sub-Saharan region. Then moving to the last two resources that I want to show you with regard to the implementation of NGS. This is an implementation manual from WHO and FIND published last year with regard to the surveillance of drug-resistant TB, but that can be adapted for the diagnostic use of the NGS and will be updated by the next year for programmatic use of NGS. The target audience are NTPs, Ministry of Health, implementing partners, and laboratory staff. And these manual address the 10 main areas of implementation of a new product, and in particular of tNGS here, with regard to the policies, budgeting, planning, regulatory, equipment, supply chains, standard operating procedures, quality assurance, recording and reporting of the NGS information, HR training, and monitoring and evaluation. These are the key steps for implementing NGS, so the need to establish a technical working group to lead the process, defining the intended use and the algorithms for tNGS, developing an implementation plan and budgets, procure the NGS materials and upgrade the infrastructures, develop standard operating procedures, including for reporting NGS data, implementing QA programs, and implementing training, mentoring, and competencies assessment programs. There are some useful annexes in these guides, and including a template for Gantt chart, checklists, budgeting, list of essential equipment, in considerations around data storage, proficiency testing, reportings, some suggested quality indicators for NGS, the terms of reference for the NGS staff, and the suggested agenda for training. Last documents that I want to share with you that has been developed by the GLI following the publication of the new recommendation on tNGS by the WHO is a frequently asked question documents on genomic sequencing for drug-resistant TB. The documents can be accessed through this link and the QR code that is reported in these slides, and provides answers to common questions that a program or a laboratory implementing NGS may have, including general questions, wet laboratory questions, questions on data analysis, and also a glossary of NGS terms. It currently includes 35 questions. It is a living document that will be updated regularly. And these are, as examples, some of the answers that you can find on the document related to general questions, laboratory questions, and analysis. And you can see that there is also the possibility to have direct links to additional resources and tools that can be interrogated for increasing the knowledge of NGS. And this was my last slides, and I want to thank you for your attention.
Thank you, Andrea, for sharing all those implementation considerations and resources. So our next speakers are from high TB burden countries that have started using tNGS for surveillance and clinical decision-making. I would like to first introduce Dr. Gulmira Kalmanbetova. She is the deputy head of the Kyrgyzstan National TB Program, and she'll be sharing her country's early experiences using tNGS. Gulmira, the floor is yours.
Okay, I would like, can you see my screen? Wayne?
It's loading now.
Okay.
Yes, we see it. It's not in presentation mode just yet.
Okay. Now it's a full screen?
Now it is, yes.
Okay. Hello, everyone. Thank you for your interest in this webinar, and thank you for organizers for inviting and this opportunity to share our country experience on the implementation of next generation sequencing in high MDR-TB burden country. Okay. As I told you before, Kyrgyzstan belongs to the WHO list with the high MDR-TB burden country. And in 2023, we detected 805 MDR-TB cases out of 4,674 TB cases totally. And I also would like to say some few words about the turnaround time, the average turnaround time regarding the releasing laboratory results. In our country, for GeneXpert TAT, it's 24 hours. As for LPA, first line 3 days, 5 days for the second line LPA, and the for phenotypic DST, 30 days. And I would like to point it out that we're also using laboratory information management system, so our results can be released quickly as soon as they will be ready and entered in this TB-MIS. Regarding the implementation of the next generation sequencing, I divided my presentation in 3 parts. And the first part is the implementation of the whole genome sequencing in our country. It was done within Stop Transmissioning TB in Hospital project funded by USAID. And within this project, we implemented 8 working packages, and duration of the project was 24 months from June 2017 till July 2019. And our partners institution is listed in this slide. It was USAID Washington, as I told you before, and Defeat TB project funded by this organization and working in the country, in our country. our supranational reference laboratory from Munich, from Gauting. And also our partners was research center from Borstel. Stefan Neymann's team was with us. And KNCV, within the Challenge TB project, supported this project in our country as well. As a result of this project, we published an article, Implementation of the Whole Genome Sequencing for Tuberculosis Diagnostic in Low Middle Income Country, High MDR-TB Burden. And in this article, you can find out information how the process of the implementation of next generation sequencing was going on in our settings. And you can find out information about what kind of equipment was procured, how much does it cost at that time, the insurance, the transportation, the shipping cost, and custom clearance. How it was done in our country as well. And of course, a lot of questions regarding the cost per sequence. At that time, we have two, which we managed to measure cost per sequence. And with the different kits, it was from 141 US dollars until 167. But from that time, we know that the prices doubled. And of course, we have to measure again how much it will cost in our country, and of course in our environment, because we have okay, later on I will tell you about the shipping. And the next project, the second part of the implementation NGS in our country, it was tNGS project funded by German government. The duration of this project, it was from the 2019 till 2022. And the goals of this study was to implement of targeted sequencing in Kyrgyz NRL, and to try to establish a collaboration between clinicians and a laboratory on the targeted sequencing to improve treatment outcome. And in this slide, you also can see the algorithm which was used during the project. And as you can see, the starting point for collecting samples and performing tNGS, it was expect them to be positive results. Here it's a few words about the tNGS project performance over the course of the 3 years, 2019-2022, with some kind of interruption in 2020 during the COVID-19 pandemic. We managed to enroll 389 patients. Out of them, 237 were male, and about 30% was female. And age of them, it was 89 years old. Of the 389 patients, we regarding the samples which provided positive culture results, it was 330 samples, and 258 had sufficient library prep quality for the sequencing. And DNA from 235 samples were sent to Borstel to analyze for the whole genome sequencing to determine the association between phenotypic and genetic antibiotic resistance results. And I would like to share with the challenges during the implementation of this project. Of course, during that time, COVID-19 pandemic time, we have 9 months stock out of reagents, which caused a significant delays for the project implementation. And I would say that we have numerous challenges that cause interruption in the supply of reagents. It was lengthy shipping times and short shelf life of reagents, and suboptimal communication and follow-up from the regional distributor. Malfunction of the Illumina sequencer in 2022 due to the damage of some spare parts. And of course, the political situation in Eastern Europe made it problematic to arrange a visit by an official representative of Illumina company Albiogen, which was based in Russia. And the lessons learned from this situation, it was that the regular technical maintenance of the equipment needs to be addressed. And as a result of these two projects implemented, the research project implemented in our country, we managed to put tNGS and whole genome sequencing in the diagnostic algorithm for the routine process of the detection TB patients in National Clinical Guideline, and it was done in December 2023. It was issued and approved by MOH. And of course, at that time, we were so happy that WHO approved finally this method, because Global Fund, which is in our country, the prime of Global Fund is UNDP, and they have very strict rules on the quality. And of course, we have to have on our hands the WHO recommendation. And in diagnostic algorithm, you can see the place of the tNGS. It's here, sorry that it's done in Russian, but directly from the sputum sample from expert MTB positive RIF-resistant cases detected by GeneXpert, the samples can be treated for the targeted tNGS. And the third part of our implementation of tNGS story is that having this technology in our hands, in having such experience, we decided that the technical assistance on the scaling up tNGS on the programmatic level is needed, and we requested USAID. And Andrea and Paola, they were in our country in February of this year and provided technical assistance that supports our national reference laboratory in setting up processes for using tNGS in routine TB clinical management, and potentially in the future, probably in the nearest future, for TB genomic surveillance purposes. And objective of this on-site visit, it was overview of the NGS workflows, how the targeted tNGS solution, WHO mutation catalog is being will be used in implementation during the implementation processes. A review of tNGS laboratory protocols, a practical demonstration of tNGS use, and development of the tNGS clinical report, and releasing them to clinicians for the initiation of the treatment of the TB patients. And of course, assessment of the current NGS results versus phenotypic DST, because we have we have to work on this as well. And of course, assessment of NTP NRL preparedness to utilize tNGS for DRTB diagnosis and surveillance. And the last objective, but not least, a definition of targets and areas of intervention for developing of costed plan of action to use tNGS in the routine DRTB clinical management and surveillance in the future. Here, the proposed diagnostic algorithm is displayed. First one, this one, it's we are now using two pilots, tNGS based diagnostic algorithm, and we are planning to perform about 500 rifampicin resistant MDR-TB samples, and a person who has screened positive for TB with the Expertum TB Rif Ultra with the positive results of rifampicin resistant, they will go to the tNGS, and now we are working on this. And of course, a culture phenotypic DST will be performed LPA first line and second line also will be taking into the consideration where the samples will be treated. After finishing this pilot project, we are going to, in routine, we are going to use the second algorithm. And we expected that within 1 year, about 200 MDR-TB samples will be treated, and about 620 isoniazid resistant cases also will be enrolled in the tNGS, because in our country the monoresistant to isoniazid is still high. And now we are using MySeq platform from Illumina. We are using version 2 runs and 13 samples per week. We are sequencing within this project. And at that moment, action plan is being developed by our partners, by Andrea and by Paola. Please. And systematic discussion of genotypic and phenotypic DST correlation is being conducted, and cross-checking of reporting of results is going on. Cross-check NGS data, for example, presence of uncharacterized uncertain mutations that might imply resistance. Of course, the cross-check of phenotypic DST quality also is being monitored and discussed, and interpretation of drug-dependent, when we are talking about suboptimal sensitivity of tNGS, targets suboptimal reliability of phenotypic DST results as well. And troubleshooting, consider retesting phenotypically with the lower level of critical results is going on. And we hope that after finishing this pilot project, we will start using on the programmatic level. And even now we are using this tNGS for the routine samples from GeneXpert positive RIF-resistant cases. And thank you for your attention, and sorry for this technical issues. Winnie, over to you.
Thank you so much, Gulmira. No problem at all. You're very clear, and could hear all of your presentation and slide. Thank you so much. Our next speaker is Dr. Sain Thi, who is a TB Technical Advisor at the National TB Program in Eswatini. Dr. Sain will be presenting on Eswatini's experience using tNGS. Dr. Sain, over to you.
Thank you. Let me upload.
So Gulmira, you may need to stop sharing your screen.
Yes, please, a second. Stop. Wow.
Just a second. Let me change to the presentation mode.
You can try clicking the icon towards the bottom.
Yeah.
There you go. Okay, looks good. Thanks.
Yeah, thank you. Yeah, good morning, good afternoon, good evening, everyone. Thank you for this opportunity. Let me go. So this is about, certainly experience of the application of the targeted sequencing result use, in the clinical management. Mainly, we focus on it. And the presentation will cover, a bit of country background and the rationale of introduction of targeted sequencing and the implementation process, interim algorithm use, and then the clinical advisory, committee, the cases where we discuss the experience about this and lesson learned and the way forward for the country. So first, let me start with the country background. It's a small country in the South Africa, inhabited by 1.2 million population. So it is one of 30 highest TB HIV burden country. TB incidence is 335 per 100,000 population. And there are MDR-TB prevalence is the 4.4 in new cases and 32% in the previously treated cases, which translated around 220, MDR-TB cases, as a burden. HIV prevalence is in the world. It's the highest HIV prevalent country, so which is 24.7%, prevalence. And in terms of TB diagnostic network, so there are 35 GeneXpert, laboratory sites where, 38 devices of the GeneXpert machine were installed. And then there are 2 culture labs. One is at NTRL, the other one is in the regional laboratory. And then we have DST, lab, including phenotypic, DST, line probe assay, and sequencing, targeted sequences, which is dplex, ISAC machine. All these are, equipped in the National Tuberculosis Reference Laboratory. And we are about to introduce Expert XDR, which is now is the verification process. So it has been installed in 13 sites, so which will be replaced in the place of LPA. So LPA will be phased out, pretty soon. So in terms of TB service organization, it is decentralized. In one, there are 151 decentralized, basic TB treatment unit, but for DRTB, it is decentralized only in 14, DRTB site. So why, targeted sequencing tNGS use was introduced it started with the story of the discovery of this RDB strain. It is, harbor with a unique, mutation, which is ApoB-I491F mutation. So in 2029 drug-resistant prevalence survey, it was, the prevalence was only 30% this strain. But in the second, DRS survey that conducted in 2018, it increased to 58%. So which, indicated that the dominance of this strain, transmission in the, population. So more complicated on that is that this strain is highly associated with beta-glucuronidase and glufosinate resistant with higher MIC, but the mechanism, of this association is unknown, okay? So this strain is not, detected by all the commercially available rapid molecular diagnostic tests, which is GeneXpert, LPA, as well as by the phenotypic DST. So as a consequence, the country faced with the high gap in RRTB, case detection, as well as suboptimal treatment for, those who would be initially diagnosed as the drug-susceptible TB. So because of all these reasons, the isotony, MOH through the NTCP has introduced this targeted sequencing, use with the support from Global Health Protection Program of German Ministry of Health through the Bela Children Foundation in Eswatini. So it was implemented as a project because by that time, the WHO, validation and endorsement on the tNGS use was not, released yet. So the project was launched in 2020, but, it was delayed. The operation of the sequencing labs, operation was delayed until November 2021 due to COVID-19 pandemic impact. So since then, tNGS results were applied for clinical management. So for the implementation of the tNGS result for the clinical management, the process that we have developed so we developed interim algorithm to guide eligible sample for the tNGS testing, and also revised guidelines and conducted trainings to the healthcare workers, of course, laboratory scientists for the laboratory aspect. Okay. So including, in the training, we also include early switch to empirical, RR-MDR-DB treatment for those isoniazid resistant cases detected by LPA and phenotypic DSD. So there were reasons about this because, as we mentioned, Eswatini is a highly the HIV prevalent country. So more than 70% of our MDR patients are, HIV, co-infected. Okay, so if we don't start, early with the optimal treatment regimen, they have very high risks of the death as well, mortality as well. So we were applying the targeted sequencing were done through the culture isolates. Because of that, there were, the turnaround time of diagnosis is pretty, long. So because of that, this empirical treatment regimen use is pretty, important for this setting. And to guide the clinicians, the Clinical Advisory Committee, which I will use as an abbreviation, CAC, was formed. And it consisted of local and international clinical, laboratory, and public health experts. Okay, so for the functionality of this committee, the TOR were developed SOP, and then, feedback templates to the health facility for the clinicians, and then also process flow, all these algorithms were laid out. So you see in the figure, on the right-hand side, this was the algorithm that layout. So the result, we have the defining of the to be efficient. So result dissemination to the clinical, from the result dissemination to the health facility, return of the form, fill up and submitted at the Clinical Advisory Committee. We define the turnaround time as the 72 hours. And then from the Clinical Advisory Committee to the clinician to feedback, so we define it as 72 hours as well. So far, we were able to achieve, that turnaround time. So this is the algorithm that we use at the interim period. Our eligible, the criteria for the targeted sequencing tests are all HR, isoniazid resistant TB cases and PDR TB cases detected by LPA or phenotypic DSD, all RR-MDR TB cases detected by genotypic DSD or GeneXpert or phenotypic DSD. So during the treatment, if there are no smear or culture conversion with the first line and second line drug, and those who fail on treatment, they are also eligible for the targeted sequencing as well. Okay. So in the algorithm, you see that the circle part is the place where the clients are eligible for the targeted sequencing test. So now I would like to share a bit about the cases that we discuss among the with the Clinical Advisory Committee. So it has been 4 years, but I'm presented here for 3 years of the cases discussed with the Clinical Advisory Committee. So we see that there was increase in 2022, but 2023 remains stable. It's mainly because the clinician with time they know that what the resistant pattern that we found are pretty similar. So as you see on the right hand side, the column bar chart, we see among those cases that we submitted that targeted sequencing detected 86% of RRTB cases, whereas the phenotypic DSD identified only 16%, and then LPA also detect only 18%, and GeneXpert also detect only 40%. So without targeted sequencing, we would miss around 70% of RRTB cases among those case discussed. So I didn't bring, overall, the overall tests, done by the targeted sequencing, because we are still, finalizing this analysis. So I didn't want to present, inaccurate data as well. So that's why, I'm presenting here only for the case discussed at the CAC. And then the case discussed in terms of their demographic and the characteristic. So age is the 38 years, but the lowest age is 14 years. And then male and female, ratio, higher, male population. HIV status, as I mentioned earlier, it's more than 70% were HIV positive. And then among the cases that we discussed, 50% were new cases who never have exposed to either, first line or second line, drugs. And then 45%, were first line drug exposure. Only 2% had the second line drug exposure. And then among those cases, beta-glucan and clofazimine resistant cases were 53%, which is highly, high proportion, I have to say. So in terms of, among those cases, got the right-hand side, table, it shows that how, fragmented our, regimen were. So as well as I like to highlight mainly for the benefit of using, targeted sequencing. So you see that this RHZE regimen, the first line drug regimen. So initial regimen, among 68% of clients, they were initially on this first line drug regimen. So after targeted sequencing result, empirically, they switch to the second line drug, right? When they receive the isoniazid resistant result from the LPA. So because of this, result, we can see a lot of patients, from the patients initially started with RHZE regimen, were switched to second line, regimen as well. And the treatment success rate was pretty good. It was, 84% out of the 37, cases in 2021 and 2022 cohort, but it's small, proportion, right? So I cannot say it's good, but anyway, it's, better than, our, when we say our, pre-XDR or, other, XDR population. But we also have difficulty in defining those cases with beta-glucan and clofazimine resistant. How do we define if they are not pre-XDR according to the WHO criteria? So we are just saying ourselves as RR-MDR-DB associated with beta-glucan and clofazimine, resistant association. Okay, so then it's also worth to note that around 22% were on beta-glucan sparing, individualized treatment regimen because of this high association of beta-glucan and clofazimine resistant as well. Okay, so then from the CAC recommendation, there were some, diverse of the recommendation about the construct of regimen as well as duration. So how do we deal with this at the country level is, clinician, they review, which regimen the patients are on, how, the patients responding on the treatment regimen. For example, clinically do well, cost of conversion is achieved or not. So based on that, they decided for example 9-month regimen or, 80 to 20 months regimen, because it's so hard to convince the clients from one regimen, to change to one regimen. For example 9-month regimen, they are on 9-month regimen. So to ask the clients to change to 80 to 20-month regimen, it's so hard to convince them, especially if they are clinically doing well as well. For 2023 cohort, we have yet to measure. But at the same time, we also introduced PPAR regimen, in 2023 February. So it's been over a year for us now. So anyhow, so it has implication for us. So as a challenge, which I will mention in there. So the benefit of the targeted sequencing U is it gives us the precise diagnosis of DITB. So because of that, we are able to provide optimal treatment regimen for our clients, as well as it's built the local capacity of the lab molecular scientists as well as clinicians as well. But the challenges what we face where it's prolonged turnaround time, right? As I say, it is used from the culture, isolate, and then we often face with culture equipment downtime because of the maintenance issue as well. And then we also need to wait for LPA result for the isoniazid resistant cases, and because it's centralized, so that's why turnaround time is pretty long. Sometimes it's more than 1 month most often, right? So that's why it caused a delay in the starting with the optimal treatment regimen as well. And then there's also low access to targeted sequencing by peripheral health facility, and because also the healthcare workers are not that much sensitized, as well as specimen transport issue as well. So then there's also delay result dissemination to the peripheral health facility. Also, regarding the treatment regimen adaptation or change, there has been related to regimen change, especially when we started on the empirical treatment regimen with PPL-M, because it's only 6 months. So there's a high incentive to both clinicians and the clients as well. So clients are frustrated with regimen change, and then also some clinicians, they want to rely more on the clinical response rather than tNGS guided clinical guided CEC recommended regimen. So having all this complexity of drug resistant pattern we have. So we also need to have access to newer drugs beyond the beta-choline and delamanid, linezolid. We also need to access to newer drugs and newer regimen. So the way to, sort of some of these challenges, now, the country has already progressed to programmatic implementation with the dissemination of the revised algorithm integrated with XPA, XDR, and tNGS, because we have already revised algorithm already approved by Lab TWG and the TBTWG group. So we just need to finalize the design and disseminate it. And then we also need to improve result dissemination through the use of electronic LIS system and the electronic, clients management information system. So which DB module is there. So if we can, disseminate the result, because now we are using email and disseminate the result to by each region, through the regional focal, DB doctor as well as regional coordinator. So that's why the result doesn't reach to the peripheral health facility. So if we step up to that mechanism, we believe that our result dissemination will be more efficient as well. We also need to sensitize healthcare worker on the revised algorithm to improve the tNGS coverage as well. And then we also need to, we are still in discussion, phase as well, our approach to empirical treatment regimen, especially for isoniazid resistant cases detected by XPA, XDR, and the phenotypic DSD as well. So let me finish with the acknowledgement to our healthcare workers who work, very hard, and the recipient of care and the community, and Ministry of Health and Global Health Protection Program, German MOH, and then Boston, a research center of Boston, who has been tirelessly, supported to us, so as well as to Baylor and other, implementing partners and the donor agency as well. Thank you so much. With this, let me finish my presentation.
Thank you so much, Dr. Sen, for sharing your experience there. So we have about 5 minutes left for Q&A. I'm sure you've noticed the Q&A box down at the bottom where you can ask your questions. Thank you to the speakers who have answered so far almost 30 of the questions. In the remaining few minutes, we'll try to ask some of the questions that may require sort of a longer answer than what can be typed. One question was about asking what equipment and reagents can be offered for next generation sequencing in terms of performance and cost. So we're planning on organizing a subsequent webinar as part of this series, where we'll be inviting the manufacturers to present on their product lines. So you'll learn all about the different equipment and reagents and the costs. One recurring question seems to be about the role of LPA as well as culture now that tNGS is being introduced. So perhaps Dr. Gulmira or Dr. Sain could comment on your perspectives with regard to how many LPAs and the need for phenotypic culture as you're going to be rolling out tNGS more and more.
May I?
Yeah, please.
For the LPA, we are planning to keep a first line LPA because, as I told during my presentation, we have a high level of isoniazid resistant cases. And for them, we are going to use LPA first line. And after that, to perform also tNGS for these patients as well. Second line LPA probably we will skip because of we will have just fluoroquinolone. Other injectable drugs are not anymore used, and we will skip. As for the culture, of course, we will have a reduced number of the culturing in our laboratory, but if we are talking about the genomic surveillance, we still need these methods for the genomic surveillance because of whole genome sequencing required the culture. So the number is being calculated, how many MG tubes we will need, etc., etc. But the number will decrease, but still keeping in diagnostic algorithm. Over to you, Wayne.
Thanks, Dr. Gulmira. Dr. Sain, did you also want to comment on that?
Yes, certainly, because the LPA is centralized, right? Then expert XDR is, we can, decentralize because of simplicity of the assay, right? We can also use as the point of care test. So as I mentioned earlier, because of the centralized, use the operation of LPA, turnaround time is long, more than 1 month. So with installation of the expert, XDR, we are going to install in 13, peripheral laboratories. So we believe that by this way, turnaround time of isoniazid resistant detection, because which is rule-in test for the targeted sequencing, right? So it's important for us, especially with this, ApoB I491F mutation, So we will, that's why I say we will phase out the use of LPA, and we will rely on the XDR for the isoniazid resistant and the other second line drugs, especially the fluoroquinolone resistant, detection as well. Even though fluoroquinolone resistant, it's pretty low in the isotonic setting. But anyhow, the LPA DNA extraction, for example, amplification. So this machine would be useful for the, even one step in the targeted sequencing, laboratory as well, because still required for the DNA extraction. Thank you. So it's not total abandonment of the LPA.
Okay, thanks a lot, Dr. Sin. There's a recurring question about interpretation of results and how they get transmitted to clinicians so that they can understand the outputs. I think Dr. Gulmira may have just answered that, in fact, but I don't know if Dr. Gulmira, if you want to comment on that, and also Dr. Andrea, I know that you're leading some work with the GLI to develop guidance on that.
Within this visit of our colleague from Milan Collaborating Center, WHO Collaborating Center, we were advised how we can ensure the quality of this targeted sequencing tNGS results. We were advised to use EQA panels, which are coming on annual basis from our supranational reference laboratory, and to use this one for the EQA for tNGS. And of course, within region or with other countries who are implemented successfully before this method, we can perform interlaboratory comparison and send some DNA for the performing tNGS in their laboratory, and or whole genome sequencing to check the results of tNGS. Over to you, Wayne.
Yeah, just a small addition on the need for an interpretation guide for the clinicians and to interact with the clinicians for having a better understanding of the tNGS report. This is actually a huge, a very important component of training while implementing programmatic use of tNGS, and clinicians should be part of these trainings. And experience in Eswatini and Kyrgyzstan as well, they represent nicely what effort has been done in this regard with the establishment of the Clinical Advisory Committee and a continuous exchange of information and discussions with clinicians when NGS reports are shared with the doctors. And there's still need likely of simple documents for helping the interpretation of mutations detected by tNGS, and also of sharing templates of NGS reports that are simple to be read and to be interpreted by clinicians that are then taking clinical decisions. And these will be made available, hopefully, in the next future. Thank you.
Thank you. Thank you, Andrea and Gulmira. So I think, yeah, as we start to wrap up, I think there's also, so there's a few things. I think that if there's any remaining answers that weren't, quick questions that were there, we'll make sure when the recording, when you get access to recording, you get all those answers are also addressed in that same link. You'll get that. And also if there's other topics that you want to hear about that are not yet, that would be of interest, even based on what you've listened to today, please either send them via email or when you, in the chat box, we'll save that. One of the things that came up is also on the performance for the different drugs and where it is today. I think one of the things we will address in the next one is very much so FIND and other groups are working on evaluations of updated versions of these assays already, because there are updates based on the mutations catalog version 2. There will be newer versions of the mutations catalog, as Dr. Mohammed mentions. There's always more knowledge that is building up as we get more data, and hence the catalog will also continue to be updated because there'll be more targets associated with resistance, which then is easy enough, in a way straightforward, to update in a targeted sequencing based assay because there's enough room to update those. Sometimes it's just a bioinformatics update. Sometimes it requires a little bit of an assay update, but it is the difference being it is not having to going back. You don't have to go back to the drawing board to create a new cartridge or a new test from scratch. You're just doing a quick update and doing a comparison reanalysis. So that's something we'll elaborate on in the next webinar alongside the manufacturer's products details. And then, yeah, so that's all mainly we wanted to highlight here. But yes, on behalf of FIND, thank you so much to all the speakers and to all of the audience and everyone for attending and your engagement and interest in all the questions.
Yeah, and from Stop TB, we also appreciate everyone's participation today, especially the speakers. We're really encouraged by the high turnout. There's still 280 people online. We're sorry we have to end this since we're already over time. And we weren't able to answer all of the questions, but as Anita said, when we send the link to the webinar recording and copies of the presentations, we're going to work with the speakers and we'll answer the final questions. So there'll be a link to all the Q&A in an email in the coming days. So as Anita said, we'll be working with together, also with USAID, UNITAID, GLI, the New Diagnostics Working Group, in planning for the next webinar. We don't have the date ready just yet, but we'll be sharing that as soon as we have it. So thanks again, everyone, and goodbye.
Thank you all. Bye.
Thank you. Bye.
Bye.